Good afternoon, everyone. My name is Suman Sinhare. I am the lead discipline scientist of soft matter at NASA Glenn Research Center. So this is the second seminar of our soft matter seminar series that we have started last March. And one of the reasons that we started this is last summer, we actually organized some workshop with soft matter researchers across US, and they really liked that format. And they said, like, hey, why don't we get a um, format or a platform where they can just come in and then they can talk about their research. So this is for
Yeah,
multiple reasons. We want to create this platform where we want to um, exchange research ideas. And as
should
some
I
of
take
you may
take
know,
I take half an hour or three-quarters? Sorry?
hi, if somebody can actually, if you are not presenting, if you can mute your speaker, that will be helpful. Yeah, so essentially, so the other, as you, some of you may know, that we have a decadal committee who are going to come out with their recommendation in summer. So we also wanted to use this platform to crystallize the community. So today we have Joe Bradley and Lawrence Wilson as a speaker, and they are going to talk about high-speed optical imaging of soft matter. I met both of them during last APS, excellent researcher, and I would say like I learned a lot personally on differential dynamic microscopy and digital holographic microscopy. They're both in UK, and Joe is in Scotland. So without much further ado, I will just hand it over to both Joe and Lawrence to take it from here. Thank you.
Thanks, Emma. So I'll just set up my slides and I'll go first. Can everyone see my screen?
Yes.
Okay, great. Yeah, so as Simon said, I'm based in University of Edinburgh. So I work in Wilson Poon's group. I'm in the fourth year of my PhD now. Today, me and Lawrence are both going to talk to you about tools which allow some really high quality measurements to be made, but using equipment that is quite general. So our goal is to shift the load from high-end specialist bulky equipment to a powerful computational analysis, so you really extract the most information you can. So I'm going to start off by telling you about a tool called differential dynamic microscopy, or DDM. In this talk, I'll give you a very general overview of the tool and talk about a few specific applications to give you an idea of what it can do. And then I'll finish up with what you actually need to do it and why it is so powerful. So before I start that, I just want to give you all a proposal. I want you all to think of your favorite dynamic system, so something that diffuses, swims, grows, anything that's non-static. So I'll give you a couple of seconds to do that. So hopefully now that you're thinking of a system, I've got kind of two questions for you. The first one is, if we use a microscope, can you see the dynamics, or do the dynamics impact the microscope image in any way? And secondly, can you describe this motion, for example, with a mean squared displacement? If the answer to these questions is yes, then this talk is definitely relevant to you, because if you can do that, you can measure it with DDM. If the answer isn't yes, chances are you can still get some information from DDM if this motion affects the microscope in some way. So if your work or anything related to it is on this list, then it means that I'm certain that the tool I'm talking about could be of relevance to you. So all the examples on here, there have been publications which have used DDM to study it. There's a huge range of systems used for, and there's also new stuff coming out quite regularly. It's still a hot topic with new people finding new applications all of the time. So if it's there, it's relevant. More generally, you might wonder, what is it we're going to measure and why? So when we come up with definitions of soft matter, usually they boil down to one or two things. Either what makes matter soft is that thermal fluctuations are strong enough to deform the material, or that the structures relax and deform on human timescales. So in both cases, it's the dynamics of these materials that make it soft matter, and therefore measuring these dynamics usually tells us something interesting. Now, when it comes to making these measurements, there are two techniques that might come to mind. The most direct way and to work in real space is to take a video of your sample and then follow these dynamics. So if you're working with colloids, you might do particle tracking and obtain like a mean squared displacement. This is great, and it can give you spatially resolved information on individual particles, which is really quite powerful. But because you have to be able to resolve the particles in these videos, you either need lots of videos or very long videos to get a good number average. Now, an alternative to this is something where you're not looking at them in real space. So, for example, dynamic light scattering. This measures many particles simultaneously and does all this average for you by default, meaning you get a good statistically average picture of your system, usually at different length scales. The issue is that this requires specialist high-end equipment, which is usually quite expensive and quite often can take up a lot of space unless you put a lot of effort into shrinking it down. So for microgravity experiments, that means you're going to need some specialist equipment that does normally one job, maybe two if you like scattering. Now, differential dynamic microscopy lets you get the same information as DLS, but it feels like particle tracking, and you're using it on the same equipment you would use for particle tracking, which is effectively a good microscope that you can get a whole range of other experiments with at the same time. The information you extract is the density autocorrelation function. If you're familiar with light scattering, it's known as the intermediate scattering function. If you're not familiar, what this effectively tells you is how long density fluctuation take to decay over a given length scale. Now, for intuition's sake, you can think of it as how long particles take to move a certain distance. In general, the autocorrelation function starts at 1, because if you're looking at a particle now and then you look at it a nanosecond later, chances are you know where it will be to within the small distances. At very high times, your autocorrelation function drops to 0, because if I look at a particle today, wait a day to see if it's diffused, I'm not going to know exactly where it will be, because once it's diffused to my micron, it's effectively a random walk. In between there, we get this decorrelation, which will depend on the length scale we're looking at. So each one of these curves on the data on the screen here is the ISF for diffusing spheres in water. Each one of the curves corresponds to a different Q, which is an inverse length scale. So effectively, a lower Q I look at, the larger length scale I'm looking at, the larger distance it's traveled. So the green line on the screen tells that the particles we're looking at take about 1/10 of a second to move about 3 microns. The blue line tells that it takes a second, maybe 2, to move about 12 microns. We can extract these timescales for the different curves and plot them kind of as a function of the length scale we're looking at. And from it, we can see that the motion we're looking at here is diffusive, because the timescale scales with the square root of the distance traveled, or timescale with mean squared displacement. In other words, what the ISF tells us is everything we want to know about the dynamics. It tells us how long it takes to move a certain distance. So it doesn't have to be diffusive. For example, if you've got swimming active colloids or swimming bacteria, you can fit an ISF to this too, and it just won't scale in the same way. If you have a model for particle motion, you can actually build an ISF for it. So from the ISF, you get particle motion in the most general sense. Now, most people know that you can get this ISF with dynamic light scattering, but I'm here trying to say that you can get the same thing with DDM using much more general equipment that's on a much smaller scale. So how exactly are you doing this? How are you getting density correlations from a video, which is what I promise you can use DDM on? So in short, images are effectively real space maps of density. If you've got a dark spot, it might mean there's more material, a light spot, less material, in the most kind of straightforward high-level overview. Therefore, if we correlate two images, we're correlating two density maps, and we get information on the density correlations. Now, this is already very cool, but then if we take the Fourier transform of these, which we can do with images quite straightforwardly with software, then we average over all of space, and instead we look at the correlations as a function of length scale. The major difference being that now our inverse length scale Q, which corresponds to a scattering angle in DLS, is a Fourier component. And because when we do a Fourier transform, we look at all of the different Q, all the different length scales simultaneously, we're doing the equivalent of many different angles in DLS. To get our different time scales, we look at different images that are separated in time by a different image. So for example, if I look at a frame now and correlate it with a frame 1 second later, that tells us about density decorrelation decays over a second. Fortunately, when we take a video, we actually get many, many pairs of images that we can correlate to extract this density autocorrelation function and get these decays as both a function of timescale and inverse length scale Q. So we take our usual real space images, we run them through this algorithm, which is usually written with, there's a lot of packages online, we use our own internal module, and this algorithm gives us tells us the ISF, which gives us the information on the dynamics of the system. Now, the really relevant thing that's true for this technique, which isn't true for other real space imaging-based techniques, is that you don't need to be able to resolve the particles in a DDM experiment. You just need to know that density maps to intensity. So with a bright field microscope, I've measured the size of colloid down to about 10 nanometers. You can also look at diffusing polymers, for example, where there might not even be explicit, resolvable, distinct particles. So based on what I've been saying, DLS and DDM are measuring the same thing, this autocorrelation function. And how do they compare in results? So I'm going to start by using particle sizing as an example, which is actually kind of my specialty and my interest in DDM. So I've shown before that you can extract a diffusion coefficient from the ISF by relating the time scales with this expression here. And if you know the diffusion coefficient, you can get the particle radius. Now that first expression on the top there tells you that if you plot the time scale against the length scale on a log-log plot, what you're going to get is just a straight line with gradient of 2 for diffusing particles. So this is shown on the screen for data taken with a dynamic light scattering setup for 3 different particle sizes. What I'm going to add to it, though, is the data taken from the same DDM on the same spheres. Now, we see 2 things from this plot. First off, we see that DLS and DDM agree well. There's the same line defining each one of the curves for both techniques for the 3 different sizes. And we also see that DDM is equivalent to doing DDLS at very low Q, which is equivalent to very low angles, which can be quite a challenge using conventional dynamic light scattering setups. Now, the major distinction here between the tools is that DDM works at far lower Q, which is equivalent to much larger length scales, and leads on nicely to my research, which is on bimodal suspensions, or generally multimodal suspensions. Now, when I say multimodal suspension, what I mean is a suspension with multiple peaks within the size distribution. So they're pretty common in reality. A good example is commercial milk, where you've got large micron-scale fat droplets and small protein micelles. Now, if you want to measure a multimodal sample with DLS, you need to carefully choose the angle you look at to avoid losing any of the modes in minima, because at certain angles, you will lose specific populations depending on their size. So if you want to measure a multimodal size distribution with DLS, you need to choose the angle carefully to avoid missing the modes. However, to make the choice of angle, you need to know the answer already. Otherwise, if you measure at 3 different angles, you get 3 different results. So what you've ended up with is a chicken and an egg problem. To know the size distribution, you need to measure a good angle. To choose a good angle, you need to know the size distribution. Now with DDM, because we work at this very low Q, this is actually not a problem. So to produce a plot on the screen, what I've done here is I've started with 2 monodispersed suspensions of spheres. I've sized them separately to get their actual radius, which is the dashed lines on the plot. And then I've mixed these 2 populations in various compositions from mostly small particles on the left to mostly large particles on the right. For each one of these compositions, I've taken some videos in bright field, just on a standard microscope, and attempted to extract both sizes simultaneously using DDM. Now, what we see is that over this wide range of compositions, you actually get straightforwardly the particle size for both populations right up until the very ends of this plot, which is where the population which is slightly biased is contributing less than 1% of the signal. Now, you could maybe get a plot like this with DLS if you choose the right angle, but to do that, as I said, you need to know the answer already, or you need to have a bulky setup that can cover a lot of angles. Now, what you struggle with even more with DLS is converting the signal fraction, so the fraction of the signal which comes from large particles and the fraction of the signal which comes from small particles, back to relative volume fraction, so back to composition, which is actually quite straightforward in DDM because, in short, the polydispersity doesn't affect this transfer in DDM, whereas in DLS, the polydispersity makes a massive difference in how much signal they will contribute. So on the plot here, I've taken the signals extracted with DDM, which is the data point, and just plotted them against a simple theoretical model, which doesn't need any input of polydispersity or angle. We know we're working at low Q, and we can see they line up really well. So in short, with DDM, you can extract composition straightforwardly from a video in a way that's not straightforward with dynamic light scattering. Now, you might have noticed here that I've said I've worked with a wide range of compositions, and I have, but the x-axis here is on a logarithmic scale. The reason for this is that the signal per particle in DDM or in light scattering scales to the 6th power of its size. What this means is that a small number of large particles can completely overwhelm the signal from small particles. This in DLS is somewhat an accepted annoyance. You can get around it by playing with the angle a bit, but it's not easy. Whereas in DDM, there are things we can do to mitigate this. So one of the major advantages of differential dynamic microscopy and the fact we record on a microscope is that at the same time as we get all this ISF information, we get a real space video. So in other words, we can take a look at this video and we can see what we're working with. We can if we can resolve a feature, we can remove it from the video before doing the analysis to get the ISF. So for a trimodal system, or a system where we've got a few large particles, we can literally zoom in between the large particles to extract the information on the smaller ones and remove the signal from the large ones. Now, to demonstrate this, I produced a trimodal suspension. So I had some micron-sized particles together with some 60 nanometer particles with an intermediate size in between. And I recorded videos of this sample, which was designed so that the large particles would completely dominate the signal in a typical light scattering or DDM experiment. Now, as anticipated, when I take the video of this and I can see these few large particles, if I just analyze it all together, I see a single peak in the diffusion coefficient distribution, which is what I plotted here. Now, bear in mind that diffusion coefficient just inverts directly to size through the Stokes-Einstein equation. So when I say you see one peak, what that means is you see one size. As I said, it was designed so that this would be a challenge, and we're overwhelming the signal from our small particles. If, however, I then zoom in and crop the video down to exclude the signal from the large particles and look between them, you can easily, what you actually extract is the size distribution of the two smaller populations with no signal from the large ones. And we extract them with enough power that we can actually resolve the two peaks. So in other words, by looking at the real space information we get and combining this with the DDM analysis, we can size three populations within a trimodal suspension that would be, quite frankly, definitely a challenge in a light scattering experiment. Now, I've shown this for sizing a trimodal system, but this actually demonstrates one of the good advantages of DDM more generally, which is that we can use this real space information directly before analysis. So say, for example, you're performing an experiment that's expensive and hard to run, say it being done on the ISS. If you get a single large colloid move into your video on DDM, you can crop it out and save most of the data without having to worry about that large particle interrupting it and without having to repeat the whole experiment. If, however, you are doing a light scattering experiment and you have the same issue, a single large aggregate moves into your scattering region, there's not a lot you can do about it. You simply have that data with the large aggregate washing out all of the signal from the smaller ones. And typically the only thing you could do is repeat it, which can be a massive pain and quite expensive. As before, when I was talking about the sizing, not only can we see the three different sizes here, but we can invert this plot and get the relative volume fractions to quite accurately. Again, all of this from bright field microscopy. So this is one of the really powerful applications of DDM, which demonstrates why it's good to work at low light scales. Now, I've demonstrated particle sizing, but that's certainly not the limit. One of the other techniques it works really well on is microrheology. So remember that I said when we do a DDM experiment, we actually extract the diffusion coefficient from the ISF. Since we know the Stokes-Einstein equation, if we know the viscosity of our suspension, we can invert it to a particle size. However, if we didn't know our viscosity and instead we knew our particle size, we could equally invert this to get the viscosity of the fluid. So in the simplest case, you can use this for Newtonian fluids. It's that same straightforward equation. And this has been used to deal with some challenging suspension. So for example, it's been used to measure the viscosity of water down to 30 degrees below freezing. Otherwise, pretty challenging. Can't do it in a rheometer, it crystallizes. Now that's true for a Newtonian fluid, but what it's showing more generally is that diffusion links particle size to the rheology. And going a step further, you can take the mean squared displacement, which I mentioned you can extract from DDM, and you can extract from this the storage and loss modulus of the material. So the data on screen is shown for some worm-like mice cells where they put in, I think, 500 nanometer trace particles. Now, what you can see on the left is that there's two distinct regions in the mean squared displacement. And this is effectively the elastic behavior of the mice cells versus the diffusive fluid behavior. On the right, they've taken this information and converted it to get the storage and loss modulus that you'd get from a rheometer. And they've compared this directly to rheology experiments. And you can see that DDM and rheology really agree quite well, with one of the major benefits being that the DDM experiment is used a sample size of probably on the order of 100 microliters, whereas a typical rheology experiment usually requires quite a lot more. On the extreme case, you can make use of this fact that you're looking at the different length scales in DDM to get a lot more information. So on the screen now is some data for a plot group which had looked at the torsioning of a gel. And what's on the plot is the time scale of the decorrelation as a function of this inverse length scale Q. So on the left, we're looking at low Q, which in other words is large length scales. We're getting an idea of the macroscopic behavior, so the overall rheology. On the right, the plot shows that the time scale behaves differently when we're looking at low Q. high Q, which is effectively on the very small scales. So at very high Q, we're looking at the behavior dynamics of the colloids within the clusters. We can see that there's 3 distinct regimes to describe the 3 different types of rheology. So low Q, you're looking at the bulk rheology, the large length scales. High Q, you're looking at the dynamics inside the cluster over very low length scales. And then you've got this intermediate Q independent regime, which is actually the dynamics of the network itself. So this entire plot on here, each line came from a single set of videos. So by taking bright field microscopy video of their sample, they could cover the dynamics in the 3 different regimes as a function of length scales simultaneously. Again, everything I've shown so far, that was just using bright field and phase contrast microscopy. But because we're working with a microscope, we can use any contrast generation method we'd like to, and each of them comes with respective advantages. So for example, if you've got a fluorescence microscope, you can dye your different populations to look at them separately. Just have a look at that. If you've got a fluorescence microscope, you can dye your populations and look at them separately. So one example that I've got on the screen here, they've got a small intruder particles in a network of very large, dense, in a dense network of very large particles, and they've tagged each population with a separate fluorescent marker. Now, before I showed that you could crop a video, and by cropping a video, you could extract the dynamics of only remaining particles. An alternative is to just look at one fluorescent channel. So if you just look at the fluorescent channel on the right on that video, then you actually extract the dynamics of the smaller intruder particles without needing to physically alter the sample. So again, using these different image generation methods lets you get a lot of the benefits that you get in other techniques. And again, earlier so far, I've mostly discussed diffusive techniques, but this is just because it's most widespread. You can also look, for example, at active particles. So your examples are Janus particles or even swimming bacteria. So in our department, we actually use DDM to sweat swimming cells quite often, although we know in reality that all cells will also be diffusing. And what you end up with is an ISF that mixes the swimming population and the diffusing population together, and you can extract information on both, depending on the length scale you look at. So on the right is the experimental ISF extracted for swimming bacteria at different length scales. So as we move along this direction, we're increasing Q, looking at smaller length scales. At low Q, we can see clearly the 2 processes. And from this, we can extract the swimming speed of the cells, which tells us about their energetics. We can extract the diffusion coefficient, which tells us about the geometry. And we can extract the fraction of cells, or the fraction of signal, which is corresponding to the fraction of cells that are swimming. So this allows us to do some really interesting physiological experiments. For example, I've used it to look at how E. coli shut down during starvation. All of this, again, coming from the same ISF. So that was just one example of a non-diffusing particle. As I've said, there's a whole range of stuff you can do. Examples that I'm not going to go into in too much depth. I've said we can do particle sizing. This includes really turbid systems, anisotropic stuff, and even protein growth, protein clusters. You can look at dynamics in some really complex geometry. So with the fluorescence experiments that I mentioned before, people have even used that to look at vesicle transport inside a cell as it's kind of going about its day without killing the cell. So in other words, even in these really complex setups, you can still get a lot of information. I've talked about cell transport. The particle sizing leads nicely on to looking at aggregating systems and the coarsening of gels and foams. You can do microrheology. So far, I demonstrated the passive microrheology, or went over it very briefly. By combining it with other setups, you can do active microrheology. You can even do DDM in kind of external fields. So you can look at people have looked at the yielding of emulsions under shear. Adroot's demonstrated that you can do particle sizing under flow as well. And while so far I've mostly talked about things that have kind of discrete trackable particles, that's absolutely not a requirement for DDM. All that you need to know is that intensity is affected by density and that you can make this change. So people have used DDM by putting polarizers in the microscope to look at liquid crystal elastic ratios, by looking at defects, even though there's nothing distinct that you could necessarily track, if that makes sense. In short, DDM should be able to do pretty much anything that DLS can do, with some caveats that I'll come back to later. So I've given a bunch of systems, and I told you earlier that if you can see it and you can model it, you can use DDM. But actually, we're all aware that no experimental technique is perfect. There's a few more specific rules. So it's all about capturing the curve that I've been talking to you about before, which means that the system must move. To measure dynamics, there must be dynamics. These dynamics must impact the camera intensity. So for example, if you just record a liquid crystal with no polarizers, you get no impact, you don't see the signal. But in polarizers, you can. The time scale of the decay must be accessible by the camera. So you're stuck at looking at images of a size you can record. If your decay happens over nanoseconds, you're not going to capture it at 100 frames a second on a camera. The process you're interested in has to be the one that causes decorrelation kind of first. So as an example with particle sizing, if you're looking at really large particles, they will diffuse very slowly. So you've got to wait quite a long time to see this diffusion over these kind of length scales. However, if you've got sedimentation, if you're doing this in a lab on Earth, and they sediment before they have time to diffuse, then actually you'll lose the intensity correlation due to sedimentation before your diffusion. So in other words, you have to be interested in the process which causes decorrelation the fastest. And finally, your dynamics have to be pretty consistent throughout a video. So you're recording these videos for usually 10 seconds to a minute. If your diffusion coefficient is tripling in that time, you're going to get something that's kind of a measured average of the lot. So your model might become a little bit more messy. So hopefully you've established thought of some systems that will work well. So what do you actually need? You need your sample of interest, an interested scientist, probably you. You need a microscope, which we've got quite lax requirements, which I'll come back to momentarily. You need a decent computer, and you need a software to do the analysis. So the microscope in general, we used quite a nice setup, which I'll come into. It has been done on the light microscopy module, which was put on the ISS in 2009, and other studies on Earth have used comparable equipment to demonstrate that you can do DDM. The microscope we use is a Nikon with a whole selection of objectives, a low noise, high frame rate camera, different image intensity modes, different imaging modes, so bright field, fluorescence, phase contrast, all fine. We've got a nice, powerful LED that can do this, and we've got an automated stage that makes all of our experiments quite straightforward. So it's a powerful piece of technology, but it's quite standard for a lot of labs. However, this isn't actually what you need to do DDM. This is more powerful than what you need. So as long as you have an objective, a camera which can do a decent frame rate, usually kind of 100 frames a second is fine. LMM had 40 frames a second. You don't necessarily need different imaging modes, but you get the different advantages. And you definitely don't need coherent illumination or a fancy stage. If you've got any of this, you can do DDM. So to really kind of drive this home, we've done DDM on a Raspberry Pi camera with a 10-pence lens, plastic lens used instead of an objective. And we got accurate size of that for particles down to 60 nanometers. So if you can beat a Raspberry Pi camera and a piece of plastic, you can do DDM. As I said, they've done it on the LLM before, and that doesn't actually even meet this criteria. So this only had 40 frames a second on their camera, but they could still get reasonable example of the slower processors. Just to finish up pretty much, I want to go over the workflow for an experiment. So most of the work you do for DDM is done kind of before and after. So before an experiment, you need to prepare your samples, plan out ahead, get ready. But the actual experiment itself, you need to put the samples under the microscope, identify some position to record and do some visual checks, make sure you can see your sample. And then you've got to set it up to record 30-second videos, which is usually quite automated. Then after all of this has happened, you typically process the data and do all the analysis later on. It's not done live. Now, what this means is that if you were to do this remotely, or you were doing it on a space station where you're requiring astronaut time that's quite restricted, most of this is actually not requiring the astronaut's input. The input time is quite minimal. So all the astronaut would need to do is prepare a sample, load it, identify the positions, and press go. All of the analysis and the time-consuming bit is usually done separately. So summarizing, effectively, this has largely been a sales pitch. So I'm going to finish off by summarizing the advantages. It shares a lot of these with DLS and particle tracking. But unlike DLS, for example, DDM is insensitive to dust or other static dirt on the capillary. That can ruin a DLS experiment, but not in particle tracking. However, in DDM, you don't need resolvable particles, so you can get very good statistics compared to something like particle tracking. You get access to multiple length scales, and I've shown that we're powerful with the gel example. And I've also shown that you can get these real images that allow more flexibility than your other reciprocal space techniques. You can use all of the different methods you've got in a microscope to have the benefits for selecting what you look at. And the sample size you actually need for a DDM experiment is tiny. You're normally talking 100 microliters. So in other words, if you've got 100 grams you're sending somewhere, you can fit a lot more samples in a DDM experiment than you could necessarily with a typical large gathering setup or definitely macroscopic rheology. All of this is done on a very general piece of equipment, which still works in every other way a microscope would do. You don't need to modify your microscope for it to work with DDM. I am also aware that we're all scientists and we know that every tool has limitations. Nothing is perfect. So in addition to the things I said required for the system to work at all, you also need to deal with some of the limitations. First off, it's not yet a widely known technique, so you typically have to explain it to people before you use it. So with things like this talk, I'm not sure if the entire audience will know what DDM is. Some of you I know have heard me talk about it before. Typically, you need to explain it a bit. The sensitivity of most cameras that you get in a microscope are lower than that you get in DLS. Now, I've seen some really impressive results where people have measured very low concentrations of protein, for example, with DDM, but I still suspect that the limits for lower concentrations will be lower with DDM than they will be with DLS. Your analysis isn't real time. It takes about 5 minutes. This can be an advantage, as I mentioned before, in terms of user time, but also means that if you want an immediate change to your experiment, it's not necessarily the ideal technique. Typically, you're looking at milliseconds for the lowest time scale. Again, you can work around this with fancy setups, but typically milliseconds. And then you've got all the same disadvantages you've got with DLS. So, for example, your large particles contribute more to the signal and will dominate it if you don't do something about it. And you only get ensemble averages, so you don't get information on individual particles. Now, inverting this can be quite challenging in the best case. And if you've got a model that you can fit to it, though, you get much more, you can extract parameters quite nicely. But if you want information on individual particles or very, very localized that isn't averaged over time and space, you need to look to other techniques. And an example of that is the holographic microscopy that Lawrence is going to talk about. So just finishing up, thank you all for listening. Think back to that system that you thought about when I asked you to think about it in the first slide. And ask the end, is there a way to watch it move? If you do, can you model this motion? And if the answer to these is yes, it should be a straightforward way to extract the same information as DLS, but on more general and often smaller equipment. Thank you very much. I'm going to pass over to Lawrence, I think, for him to take over and talk about DHM. Thanks.
Thanks very much, Joe. Can you all hear me okay?
Yep.
Okay, that's great. Thanks. I'll just share my slides. Thanks very much for that introduction, Joe. Okay, and hopefully you can now see the slides I'm presenting. So please do tell me if you can't see them, and I'll endeavour to do something about that. So we're running a little bit behind. I'm going to move along quite quickly. So the technique I'm going to talk about, holographic microscopy, it lets you image things in real space. So as opposed to DDM, you can image things in real space and in 3D. And this kind of cartoon that I've got here is an example of swimming tracks of E. coli in a small sample chamber. And I'll tell you a bit about some of the projects, some of the applications we've found for this, and a little bit about how the technique works. But this is mainly going to be some references that you can look at, where you can drop me an email afterwards. I'm not going to get into the weeds on exactly how this thing works. I'm just going to kind of show you the sort of things it can do. So my lab here in the University of York was originally a soft matter physics lab. So to me, a bacterium is just a swimming colloid. I was talking to Suman about that at the beginning of the meeting. And again, the idea is to shift the heavy lifting from the hardware into the software, so we can work with relatively straightforward subjects. My lab's looked at swimming microbes for most of the time. The interest there, the crossover with soft matter, as well as existing on the same length scale as colloids, with the interaction between microbial cells and soft matter systems like polymers or liquid crystals and that sort of thing, is also of interest. So this is a holographic microscope. This is what it looks like. It's based on a simple simple inverted microscope in our lab, but we replaced the light source with something that's a bit more coherent, so either a laser or an LED. And the picture at the bottom in the middle is what you get when you switch the camera on. This is a suspension of bacteria, and of course the background there, you're seeing a lot of diffraction fringes and things and scattered light from objects in the optical train. Of course this stuff is all static, so you can perform a background subtraction, and what you're left with is a picture like you see on the bottom right, and each set of diffraction rings there corresponds to a bacterial cell, which is either closer or further from the camera's plane, depending upon whether its size is small or large. And this image here is about 300 to 400 microns on the side. So in order to get some information out of this this holographic data, you can do the full Maxwell equations. You know how light scatters from simple objects like spheres, and you can solve some kind of nasty looking equations to fit the scattering pattern as detected by the camera. And David Greer's group in NYU did a whole load of sort of pioneering work on this maybe about 15 years ago. Vinny Manoharan at Harvard has done some bits and pieces too, looking at colloidal systems and colloidal clusters. We take a slightly different approach in my lab, which is more of a heuristic approach. So biological samples obviously come in lots of different shapes and sizes, and we often don't have an analytical model for what we expect the scattered light to look like. So people familiar with DLS, dynamic light scattering, as Joe was addressing before, might be familiar with this idea of a Rayleigh-Gans scattering regime. And that says that if you have a weakly scattering object, you can divide it into small independent scattering centers. So instead of looking at a long fibrous object, like the thing I've got on my screen here on the right-hand side, I've got a long blue thread, and I can consider this thread to be a chain of independent scattering centers, and they scatter light, and the unscattered and the scattered light is collected at the image plane of the camera, and we can decode the information that's there without any assumptions about the shape of the object. So to give you some ballpark figures of what we can do with this kind of method, we can track hundreds or thousands of cells simultaneously in 3D. And unlike DDM, this is a tracking method. So we do have to resolve the objects or at least see the scattering pattern in some sort of way, in a way that we can track. So we can image volumes at between 30 and 2000 volumes per second. Those are the typical rates we use. That's simply limited by the camera. So each 2D image we get from the camera contains 3D information about the sample. The resolution is similar to bright field optical microscopy. It's something like diffraction limited, but of course you can do clever things like fitting centroids of blobs and that sort of thing if you're interested in finding the centroid coordinate of something. And in a similar way to the way that we look at super resolution fluorescence data, we can kind of improve the resolution a little bit. It's not strictly diffraction limited depending upon what you want to do. And we have sensitive volumes that's something like up to 1 millimeter cubed. So this is the technique that we use to reconstruct the sample volume. At the bottom left-hand corner, I've got an example of the image that I showed earlier on, and it's got these ripple patterns caused by bacteria that were swimming around in a liquid sample. And these could equally be colloids or anything else. It doesn't have to be bacterial samples. David Greer's group in NYU, as I said, started a lot of this work looking at characterizing colloidal particles, but I'm interested in tracking bacterial cells. The center is a kind of cartoon of how we go about interpreting the holographic data. So we take our raw frame and we apply an algorithm which refocuses that image at different axial distances from the original. And by that we can build up a stack of images, and this stack of images is like refocusing the microscope image, but purely numerically. So you only need one raw image in order to capture the 3D information. And when you've done that, you can find out where the particles are in the 3D volume at that instant in time. You can go further than that and you can say, okay, I've got one single frame and I can see where the particles lie in my single frame. And if I follow objects through time in the same volume, I can build up tracks of particles or tracks of cells, and we can see what they're doing. We can understand something about the dynamics, how they interact with each other, how they interact with the walls of the sample chamber. And because we don't rely on averaging throughout the whole time series, we can do things like add a stimulus and then see what happens next. I'll give you a quick introduction to some of the projects that we've looked at in the past. We started out by working on motile eukaryotes. So these are cells that have a nucleus in them. They're colloidal in size typically, so between a few hundred nanometers and a few micrometers. On the left-hand side, we were looking at the waveforms of swimming malaria parasites. In the middle, we tracked some sea urchin sperm as they were doing chemical sensing, and we were trying to understand the mechanisms by which they can sense their environment. This is a classic model system in the field. And in the bottom right hand corner, this is one of the latest things from the end of last year. This is a paper that's on archive at the moment, and we've been imaging the flagella, these kind of beating organelles at the front of the cell, from Chlamydomonas reinhardtii. We've been able to image that with sort of a couple of 100 nanometer resolution, and we've been imaging at 800 frames a second. And this is an important model organism for studies of conditions in humans like cystic fibrosis and infertility and different things there. I wanted to focus on today 2 examples that are probably closer to colloidal science and soft matter, because I think this is more interesting to the community. I'm going to talk about flagellar wrapping in a bacterium called Shewanella putrefaciens. This is in collaboration with Kai Thormann from Gießen in Germany. And in the second example, I'm going to talk about archaea, which are an interesting third branch of the tree of life. So aside from bacteria and eukaryotes, and again, eukaryotes are sort of plants, animals, fungi, that sort of thing. The archaea are a distinct third branch that some people claim are ancestors to the other 2 branches. That's a controversial topic, but the interesting thing to me is that each of these branches of the tree of life, the bacteria, the eukaryotes, and the archaea, have evolved their own way of moving around. So we're looking at the way that life solves the problem of colloidal transport, and it's arrived at 3 different solutions from different starting points, which show interesting similarities. I wanted to highlight at the bottom here as well, we also work on active colloidal systems. I think this could be a very interesting system for low gravity and microgravity environments, because in these situations you've not just got a gravitational force, you've also got a gravitational torque on a bottom heavy particle, and that's the sort of thing you can't density match out. But we did some experiments using holographic microscopy to try and understand the dynamics and the flow fields around these particles. So the first example, and I hope you can see, I'll enable my laser pointer so hopefully you can see the image in the top right hand corner here. This was an observation by my colleague Marco Kuhn, who's working with Kai Thormann in Gießen in Germany, and he'd labelled the tail of a bacterium. And you can see in the central panel there's a bacterium at the bottom, and at the back of the bacterium there's a long helical tail. The cell body is about 2 microns long and the helical tail is about 10 microns long. And in the top panel you can't see the cell body, but it's at the top right hand corner. The cell body isn't labelled, and to the bottom left of this panel, you can see a kind of helical shape. And what you're going to observe is a bacterium that's trapped in an agar pocket, and the tail's kind of spinning around and flapping from side to side. And then something interesting happens. As I play this, you'll be able to see. So the tail's flapping from side to side, and then suddenly it pops, wraps around the cell body, and this allows the cell to escape from its entrapped location. So let's just run through this again. So the tail's flapping side to side, and it suddenly pops back. The whole structure reverses, wraps around the cell body, and allows the cell to screw out of its location. So our contribution to this was to look at what happens as these cells swim in different viscoelastic environments. And it turns out, I'll let you read the paper if you're interested in the details, that the tail of the bacterium is made out of 2 mechanically different components. There's a stiff component next to the cell body and a kind of floppy component further out. And the biologists can do clever things to interrogate the physics here. So, for example, they can swap these 2 components of the flagella around to see what happens. And we can investigate why these cells bother having such a sophisticated architecture in their flagellum. It turns out that this particular architecture is critical to allow them to navigate in very viscous or highly structured environments. And what you can see in the bottom left-hand corner of this slide are 2 tracks. So the kind of purple track, the bottom left-hand corner, is cells swimming in a fairly heavy fecal solution. It's a viscous environment. And on the right-hand side, we've got the cells swimming in a much lower viscosity environment. And I've got this kind of paintbrush style plot whereby when the cell moves fast, the lines are thin, and when the cell stops or reorients, the line becomes thick to indicate that the cell's speed is slow. So in this situation where you've got a water-like Newtonian physical environment, the cell swims fast, reorients, swims fast, reorients. In the presence of a viscous agent, the cell spends a long time swimming relatively slowly, and this is because we're able to trigger the cell flagella wrapping behavior. But being able to observe the cells swimming in 3 dimensions gave really important insight into the statistics of how often these flagella were wrapping around the cell body, and more information about the physical constraints necessary to produce this behavior. Again, the reference to that paper is at the bottom. It'll be on the last slide as well that I'll leave up at the end. The second case study I wanted to talk about was to do with these archaea, these sort of what are thought of as primordial organisms, and they live in interesting places. They seem to have been out-competed by eukaryotes and bacteria in a lot of environments. They're very prevalent in harsh environments. So we went into the wild to collect some of these organisms, and you can see the picture at the top with this pink lake. That's Great Salt Lake in Utah. The lake goes pink because of the abundance of these salt-loving archaea and other species at certain times of year. So in collaboration with Bonnie Baxter and Jamie Butler from the Great Salt Lake Institute, we isolated some archaea from here. We also isolated some archaea living on the picture on the right hand side. This is a dark matter lab. It's the deepest working lab in Europe, deepest working mine, sorry, in Europe, and it happens to have a lab in it. So we can go down and sort of capture and isolate these interesting organisms that creep out of the walls. And again, this picture in the middle shows you our home depot constructed microscope. So we have got a cordless drill and a battery there that powers everything. We've got a nice table from REI, the outdoor store, and a little laptop there. And we're able to go and do some of these experiments outdoors. So it is a relatively unchallenging technique, experimentally speaking, as long as you get a couple of key ingredients right. And the thing we were most interested in is how these cells, these archaeal cells that we've identified from Great Salt Lake and from Bulby Mine, how they would overcome rotational Brownian motion and be able to navigate up a chemical gradient, so doing what we call chemotaxis. And these cells are interesting because they live in very nutrient poor environments. They live in extreme conditions. It's a harsh, salty environment where nutrients are very poor, and they're not abundant in the slightest, and they're often ephemeral, so they appear for a while and then go away again. But because not many organisms live in these environments, there's not a huge amount of competition. And so the only thing the cells are competing with really is Brownian motion and physical laws. So we're seeing, in essence, the most efficient way that a cell can navigate chemical gradients in a nutrient poor environment. And to try and understand this behavior, we coupled our DHM experiments, our holographic microscopy experiments, with some numerical simulations. The nice thing about DHM is we can track our cells in 3D. And in the top right hand corner, you've got a little vignette of a cell swimming. It swims using a rotary motor, and that manifests as a kind of slight corkscrew character in the trajectory of the cell. But by doing some fitting, some model-based fitting of these trajectories, we can very accurately extract rotational diffusion, which is what we need in order to build up models. And we've got some details of the interior chemical signaling, internal chemical signaling structure. I'm not going to go into that here because there's not really time. I just want to go through to show you the power of this technique in determining physical parameters for a system that then lets you run simulations. But instead of having an experiment that runs for a day, pardon me, or an hour, we can use numerical simulation to say, okay, well, these cells are living isolated in their environment, and perhaps they're there for weeks, months, years at a time. And we can use that to probe the ultimate physical limits of chemotaxis in harsh environments. So that was a second paper that was published in 2020. I'll put the details up towards the end. But one thing I wanted to highlight is that DDM and DHM are really nicely compatible. So this is an example of a microscope modified to do some holographic microscopy. So holography is a little bit fussier than DDM about its setup details. DDM allows you to do things like size particles. So Joe was explaining this before. So this is particle size as a function of scattering vector, and we were able to detect the particle size from DHM data, but using DDM software. So from the same raw data set, you can reconstruct particle trajectories and size individual particles, and you can also get the collective response through DDM. And these two approaches can inform one another, and you only have to take a single set of video data in order to do both techniques. Lastly, I wanted to mention that AI and machine learning, so as the entire world is now looking at AI and machine learning, instead of going through this complicated reconstruction procedure by taking an image and then reconstructing it and then using some object recognition software to determine the positions of objects, you can simply feed in the analyzed data set, feed in the raw data, and you can train a neural network to look straight into the hologram to extract the coordinates of cells. And we've done this using, this is an NVIDIA Jetson over on the right hand side. We've trained an NVIDIA Jetson to do imaging, and we're doing sort of real-time tracking of cells and particles in 3D. So we can image cells at about 13 to 20 frames per second, depending upon the size of the image. But we're getting real-time imaging and feedback from colloidal and bacterial systems. So with that, I wanted to leave you with the punchline to say, hopefully we've come in just about on time. So the punchline here is that holographic microscopy lets us measure the dynamics of colloidal particles in 3D and in real time. And there are a few references just down at the bottom there in case you were curious to see more about what we've used this technique for in the past and the kind of apparatus we use to do it. It's very simple. And I thank my collaborators in the bottom left hand corner, and thanks for your attention. What I'll do now is I'll hand back over to Suman, and happy to take any questions as well.
So that's an awesome talk from both of you. And I will take this moment to actually to announce our next speaker. Let me share my screen. Our next talk will be on 12th of April at 12 p.m. Eastern Time. Our next speaker will be Dr. Aishman Sen, and he's going to talk about active matter swarms for cargo capture, transport, and delivery. You most probably have gotten the invite. If you did not, just let me know. I will definitely send that invite to you. So having said that, so thank you, Lawrence and Joe. Thanks a lot. This was again, I mean, I just want to say one thing to the audience is, I learned about these techniques last year, and over last, I think almost 1 year, I have had numerous conversation with both Joe and Lawrence. They have been extremely helpful. We are actually looking into these two methods of how we can use for soft matter, especially colloidal particle characterization. So I felt that the community need to know about this process. So with this, there are some questions. Patita, you have your hands up. So if you have a question, just put your hands up or just speak up. That should be okay. Thank you, Patita. Go ahead.
Yeah, thanks thanks for
Yeah.
Lawrence and Joe. Appreciate it. I learned. And I've been working with Suman on this thing for almost like a year, and then we start working on this thing. Now, there's a question for you: is that is there some kind of like a theoretical limit on your DDM? Is it limited by the instrument function, or is it limited by ISF that you cannot go? How small can you go for particle sizing? Yeah, one that would be more or less to Joe. Okay, and then for him also is that for DDM is you're more or less, as you explained through, is for the translational diffusion constants that you're tracking into it, right? And whereas it seems like Lawrence's work on the DHM is it's on the rotational diffusion constants kind of thing. Am I correct?
So I'll start with first questions, probably easiest, and come back on the second. So if I put these up, I think the major limit in terms of size is actually this. I seem to be able to click things. Okay, so this slide here, theoretical limit for lower size. I would say it's more at the moment an equipment limitation than it would be them reaching the theoretical limit. So theoretically, I'm not convinced there is one, but practically, you're going to need to be able to access the timescales that are getting very low. As you get to smaller particles, you've got a very high diffusion coefficient. So say, for example, you're looking at 30 nanometer particles in water. At this point, you're already looking at diffusion coefficients, which can get very high. So you're already kind of on the millisecond scale for a particle to diffuse a few pixels in the image or across very large distances. So in other words, if your particle diffuses more than a few pixels between frames, you already lose the information that happens in that time. So your limit for lower particle size mostly comes from the limit on the frame rate. So the lowest I personally have done is 10 nanometer radius. I've seen some unpublished data where they said they've gotten the size of proteins that were kind of 5 nanometer radius of gyration. But once you get to very small particles, your signal becomes very weak because they don't scatter much light or they don't necessarily present as much of a signal to your intensity. So again, you've got a challenge of kind of identifying these particles and be able to track the signal from them, being able to get enough of an impact on the intensity to get a signal. Does that answer the question?
Yes, very good. And then as well as to both of you, I mean, before you jump into the rotational and translation, with this kind of like a particle tracking, I'm pretty sure either your group or somebody might have done the PIV. Have anybody?
Yeah.
Okay, because it looks like, because see, yours is like it's it's not real time, because see, there is a processing time for it. So if there is an approach that one can process, what should I say, parallel processing, doing one section, and then another computer or whatever is doing another section, and so on, and then combined together, probably through AI. I don't know. Can we do PIV?
Well, I mean, the holographic microscopy is kind of, I guess it's fundamentally different in some ways because we don't require the dynamics to be stable. So we can take one frame at a time and watch the time course, but then I guess we lose the kind of the ensemble averaging and the sensitivity of DDM. So I guess I mean, I'm not sure what Joe and the rest of the guys in Edinburgh have been doing, but I mean, we do a little bit of that here. But sorry, Joe, continue.
Yeah, well, we have done a little bit of PIV. The challenge with PIV is it's not something I've done. I just know people in the group have looked at it. From what I understand about PIV, you're normally looking at kind of a collective motion. You want to describe the collective motion, but that includes direction. So you can use PIV, for example, to measure a flow speed, because you've got your collective motion from left to right, for example, that works quite well. Whereas DDM, you don't need a collective motion. So if you try to use PIV, again, my understanding, I'm not fully following it, just in the most straightforward way on diffusing particles, you'd still get a signal because you've got some moving left, some moving right, up, down, all directions. So I believe some people have combined PIV. So for example, they've used PIV to get a flow rate, which they've then corrected to do DDM. So it's a powerful kind of complementary technique, but I wouldn't describe it as a good alternative. If you do have collective motion, DDM you lose some of the advantages of DDM that DPIV could help for.
Yeah, I agree. So Parita, just hold on. Emma, you have a question. Please go ahead.
Hi, very nice talk. Thanks for sharing. I have two questions. The first is to Joe, and I want to just confirm that DDM technique only mirrors homogeneous movement. Is this correct? Because sometimes swarming behaviors, they may be heterogeneous. We may find some error, the particles behave one way, and some error particles behave another way. Mm-hmm.
Well, to some sense. So for a good example for that is if you remember the video I showed you where I cropped the video down and looked at the region between the large particles.
Oh.
I average everything effectively within that cropped region. But if instead of cropping, you were to break your video down into 24 segments, I don't know why I said 24, odd number, 25 segments, say a 5x5 grid, and analyze each of them separate, you can get some spatial information.
I got it. Mm-hmm.
But within those regions, you get an average.
I see. And thank you very much. And the second question is for Lawrence. So at the end of your talk, you mentioned about combination with AI and machine learning. The holographic technique is able to resolve real-time motion in 3D. Would you please elaborate on that a little bit? Mm-hmm.
Yeah, by all means. So in the past, we've taken these holographic movies and what you get is that kind of fuzzy looking image, and you do the background subtraction and you feed it through an algorithm. For each frame, it gives you a set of 3D coordinates, sort of just literally a list in text of X, Y, Z in microns. But that actually constitutes a training set for data. So you've got your raw image and you've got your processed coordinates, and you can just feed simple off-the-shelf things like YOLO, V5, and all these other kind of fairly basic image recognition things. You can just train them to look directly into the holography and say, okay, this is how far away that particle is. And these little Jetson computers can do that at sort of 13, 15 frames a second.
Okay, well, thank you very much. It's very, very helpful. Oh, by the way, I just realized I have one more question for Joe. And you mentioned about advantages and disadvantages of the DDM technique. I actually wrote down notes. Would you please just elaborate a little bit about this?
Yeah, sorry, was it a specific advantage or just
Yes,
talk about them in a bit more?
the advantage and disadvantage of DDM.
Yeah, so, well, I mean, describing this is always relative to other techniques. So the examples I talk about a lot are particle sizing, particle tracking, and DLS, because these are kind of the most directly relevant. So relative to particle tracking, you've got really high number densities sometimes because you don't need to be able to resolve the particles. Relative to DLS, the general idea is that it's much more robust. So if you have something go wrong with DDM, there's more of a chance that you'll be able to deal with it. So a piece of dust on a capillary doesn't affect DDM. DLS, it throws off a lot of your measurements. A lot of it is flexibility and that the equipment is just very general. The disadvantages usually come down to kind of sensitivity and timescales. So the ones I put up were on here. The disadvantages that are unique to DDM, well, again, unique compared to other techniques. So it suffers from a lot of the disadvantages that DLS do. One of the major examples being converting from the ISF back to standard information. So before the talk, I was chatting a little bit about how you go back to a particle size distribution. That relies on some assumptions if you don't have a model that you want to fit to it. For example, converting from the ISF, trying to work out whether you've got one broad polydispersed thing or two separate populations that are monodispersed is not straightforward. Whereas with particle tracking, you might be able to see that more directly. Does that all make sense?
Yeah. Thank you very much. I got it. Thank you very much. I really appreciate the talks.
Thank you, Emma. I think Bill has a question. Bill?
Right, first I want to thank you for the talks. They're very nice. We do dynamic light scattering now. We do it with teeny little boxes, and we see a lot of the same things. But one of the things that concerns me is when we look at really small particles and we want to look in the backscatter so we don't have to worry about concentration effects, we go to 100 nanoseconds or microsecond timescales, which I don't think we're going to find a camera that does that. So does that imply that most of your measurements are made in forward scatter? You use that kind of?
So it's slightly different in that. So DDM often it's spoken about in the literature as a scattering technique, but you don't need it to be scattering. So, for example, you consider the fluorescence setup. There's no light scattered there. It's solely light that's coming from the particles. It's not coherent, so on and so forth. So there is a distinction. While the Q that we use in DDM and the Q we use in scattering are interchangeable generally, they aren't the same thing. But effectively, also in DDM, you are almost always working at very low Q. You never work at the high Q that's equivalent to backscatter of 180.
So you forward scatter and you worry about mean scattering. Okay.
So Joe, if I like from a non-optics guy like me, from my perspective, I think you mentioned in your slide, which is this is an intensity map variation. So you are just measuring the intensity variation. So if we think of that perspective, I think it's a much simpler way of thinking. Like as long as I have an intensity variation, and in this case, if it's a Brownian motion dependent, it's a Brownian motion dependent. But if there are like other flow and other things, that's a different case. But in this case, this is essentially an intensity map variation. That's how I see this. Is that a right way of thinking?
I personally prefer that. So yes, quite often. So the thing with DDM and a lot of the early literature, they were using a coherent light source, and it was very carefully set up so that they were effectively doing heterodyne scattering and working at forward scattering. Whereas once we kind of understood it more, people looked into it more, we stopped using coherent light sources. We no longer need to think of it in a scattering perspective, and a lot of the maths is a lot more straightforward and flops out more nicely if you looked at it from the perspective you're talking about of just images or intensity density maps.
So it's like an intensity autocorrelation function in the same way that it uses the Friedholm equation, and it's the same in the. So I mean, you have all. It's very much like dynamic light scattering, from what I can see. Yeah. Okay. I wanted to ask Joel a question. I was reading some of your papers you sent. Thank you very much for that. And so I looked at the images. One of your papers, you mentioned that you were taking multiple depth images. Was that because the numerical aperture of the lens made it very short and you wanted a bigger volume? I mean, in your talk, you said you just take one snapshot and you get everything. In the paper, I think you mentioned that you were doing multiple exposures. I take it there's two different approaches depending on what you want to do, depending on the numerical aperture of the lens. And really, this leads to the question of how concentrated a system can you get for whatever size particles. If you're, I mean, some of the stuff is way out of bounds, I think, for DHM. Like when we look at 62% concentrations to see glasses crystallize in space. I'm assuming that would be really tough, but I'd love to hear your thoughts on it.
You said, Joe, was that to me?
Yes, please, yeah, John. Lawrence, I said Joe, I meant Lawrence, I'm sorry.
Sure, no problem. So, yeah, so by 62%, you're going to get quite, it's going to be quite difficult to image, but I think that's true for DLS as well, right? I mean, you play lots of tricks.
Well, we don't do DLS with that. Yeah, we have to do it. We've been using confocal for that.
Yeah. So I mean, I'm really interested to see what would happen if you started playing some of the tricks with refractive index matching that people do. So high refractive index tracers in index.
We have a lot of that data. We could probably share that with you. I mean, the data is we match it to like 3 or 4 decimal points, so we can see. We had to see like several hundred layers into the sample to determine whether it was FCC or RCP. I mean, we wanted to say what's the most fundamental crystal when you form a glass in space, and we're about to publish it. It's FCC. And it's a single crystal. I mean, it's one crystal the size of the capillary, which is what was amazing to us. Absolutely. Thank you for a very nice talk, both of you.
So, yeah, I think Carolina, you have your hand up.
Yeah, can you hear me?
Yes, it's a little muffled, but it's okay. Go ahead.
Okay. Yeah, I'm curious about how gravity would affect the predictions, because I'm aware of how in past biomolecular condensate experiments, for example, done by Stanford, I think in worms, they realized that changing gravity, or like the calculation of gravity, affects the pressure involved, which changes how the condensates behave, which has to do with the movement and how they condense, come together or disperse. So I'm just curious if, we're doing this in space eventually, would it kind of mess with the predictions?
I think that's a really good question. I mean, I guess it depends upon what the most important fields are. It sounds like in this, I'm not familiar with this particular experiment, but it sounds like gravity was an important sort of control field for establishing the condensate. Is that right?
Yeah. I mean, it depends on the biomolecular condensate, which is just basically just different types of proteins.
So yeah, but I think Carolina, you are breaking out. So maybe let me simplify the question with what I understood, Lawrence. Lawrence and Joe, what she is saying, like with all the algorithm and the processing that you guys are using, so you don't see the gravitation part over there, but obviously it's a factor. But how that is, if and how that is going to change if you go in a different gravity condition, or you think that the processes are pretty much like gravity independent. I think, Joe, you kind of briefly mentioned, like, if the things are sedimenting, this will not work. I think along that line, that was the question.
Yeah, I think so, at least for DDM, everything. So at the moment, we haven't used any machine learning, and we've nowhere specified gravity or anything similar. So it's not trained on stuff that's used gravity, or in the algorithm doesn't rely on it. So the technique itself should still work very much the same. You will measure the dynamics as they are in low gravity. The difference may be the models that you use to describe those dynamics, so how you fit the data there. So, for example, you may no longer need to account for the fact that large particles will have sedimented. Or if you were looking at some fluid that for some reason had a viscosity dependent on pressure, it would change how you got particle size from diffusion coefficient. I'm making that up. I don't know how common that is. My point is, though, that what you measured would be the dynamics as they are in that setup, but the specifics of the kind of inversion of that back to useful information will depend a lot on the system, and there you may have impacts from gravity. But I'd imagine that's probably what you're trying to study if you're doing the experiment in those conditions.
Okay. Pradita, you have a question, please go ahead. You are muted if you are speaking, Pradita.
Sorry, thanks. Okay. It's more or less towards like a Joe. There one graph that you show about the what you call it like a multimodal suspension, and in there, okay, you have pretty much a distance like starting from like one is like on the far end like a 60 nanometer, and then you went to 240, and then to say like 1 microns. Okay, now that's fine. All right, now what would be the limit that you, with the DDM, that we will be able to distinguish between particle size, say like 60 to 80 nanometer or 60 to 65 nanometers?
So
how do we overcome that?
Is you asking how close the sizes would be before we'd have a challenge?
Yeah.
Okay. So there
would we overcome it? Yeah.
Well, I mean, there certainly is going to be a limit practically. So in my experience, going down to a factor of kind of 2 in size distinction, so looking at 500 nanometers versus 250 nanometers, if you try and measure them both simultaneously, you tend to see kind of one broad peak if you try and invert it, because this inversion is not straightforward. It's kind of it's an ill-conditioned problem. You have to use all kind of challenging algorithm to deal with it. And I actually use the same algorithms that we use in DLS. Even if I fit a least bimodal model to it, so explicitly say there are 2 populations here, give me the sizes. If they're within a factor of 2, I tend to see a bit of a bias, where that exact value depends on polydispersity. However, if you're measuring the populations separately, so say you have a population of 80 nanometers in one suspension, and another suspension with a suspension of 60 nanometers, you can easily distinguish those. The problem is that when you combine them, it becomes very difficult to distinguish between 2 close together suspensions and 1 broad size distribution. The exact limit, I couldn't tell you off the top of my head. It depends on the system.
Yeah, but I think we can talk offline. In my experience in my experience, what I have found out, like this ISF function that Joe was mentioning, it becomes like really difficult to convolute. But that's a different conversation. We can talk more in detail. But Lawrence, I have a question for you. There was something really interesting that you showed with regard to DHM, which was the Salt Lake. Right next to Salt Lake, you had a picture of where you basically said, like, I took this camera system from REI, we just drilled it on a system. And so what it means is like it's extremely robust. And my understanding is you don't have a laser there. You have most probably some LED, and it's most probably an inline. I mean, for the people who do not know, there are 2 processes, inline and offline. I think what Lawrence is showing here, this is a off-axis, not offline. What Lawrence is showing here is inline, meaning everything is in the same direction. So how robust it was, like, and how you were able to put that together, I would say. And do you see, when you take the result, and do you see a difference of that versus an off-axis? So that is first part of the question. The second part of the question is, with DHM, what is your limit theoretically of the size particles that you can see? You can change the order, maybe start with the last one, and then we can go to the second one. These are like really 2 important questions that I had.
Yeah, sure. So I think the size limit depends upon the amount of light that the particle scatters. I guess it's like a lot of different processes, and the ultimate limit that we've measured, 20, 50 nanometer gold spheres, but of course they scatter light like crazy. And the big limit that you hit with the on-axis configuration is that you've got the unscattered beam passing through the sample in the same direction as the scattered radiation. So if the scattered radiation scatters a fraction which is smaller than the bit depth your camera can see, then you're done. So you can, with the off-axis configuration, we don't tend to use that because we find it's less robust. I know some of my colleagues in the UK have had a go at building these things, and it seems the trade-off, if you're going to go out into the fields, you kind of want the on-axis thing, because of course, if you've got a common path in the scattering and the experiment starts to shake or move a little bit, it kind of makes your life a lot easier if it's on-axis compared to off-axis, where the two arms can shake independently of one another. So in terms of the size limit, yeah, there are probably tricks you can play to improve that a little bit, but at the moment we're hitting probably 20 to 50 nanometers for gold particles.
I mean, with an inline or on-axis system, right? And so,
Yes.
and with regard to your with regard to the Salt Lake system that you were talking about, you did not have to use, I presume, a laser. You just used like a decent LED. Am I correct?
That's right. So for that and for our early work on malaria samples and that sort of thing, we can use an LED, but if we stop down the numerical aperture of the optical system, you get something which has kind of got that little bit of spatial coherence. And as soon as you start to get the diffraction fringes in your image, basically, then you're in business.
So Bill, you have a question.
Yeah, I'd like to ask. So when people say that you can do 3D imaging off-axis and on-axis, it seems like they're saying different things. It seems like 3D on-axis means you're just seeing the front of the things and how deep they are, right? Whereas off-axis, you're actually seeing 3D because you're painting curved stripes across your stuff, right? So
I think that's right. Yeah. So it's the reference wave which comes in off axis.
Yeah. And if we had a way to make it so that it wasn't vibration sensitive, there might be advantages to off axis, because I mean, Padeth and I have built fiber optic systems that self-align. So that might take care of the problems of having the arms move and having it be really big, because you can just wind the fiber up and spool it around a spooler to strip the cladding modes. So I mean, we're thinking about that. And I mean, that's something Padeth has published in the past. You know, we did it together, but I did more of the math and he did more of the fiber stuff. So I think, do you see any problems with that approach? I mean, before we invest lots of time in it, I mean, we're close, but you've got a lot of expertise and we're just catching up. And we have other expertise, but I'd love to hear your thoughts.
Certainly from the illumination point of view, I think that's really compelling. And the thing that has always intrigued me about the off-axis system is that you can tune the intensity of the reference. And that's going to really push down the floor of the size of things you can see. So you're going to get down to. Yeah.
And our fiber splitters are variable ratio. You can there's a knob on them, so you can okay. And that was the big problem we had when we first started, was we found we had to keep changing the neutral density filters, and we had to run to different labs to find the right ones.
Yeah, I see. It's nice to have a dial you can turn there. Yeah.
Okay.
That's awesome. So any more question from the audience?
I just have one more. So Joe,
Yeah,
let me ask a somewhat interesting question. As far as you were showing this Stokes-Einstein equation, which assumes a spherical chicken. But I assume that works for most everything because you're just looking at intensity. So you don't really have to worry about the models, right? You don't have to worry about different kinds of models outside of the Stokes-Einstein, or do you?
In what sense do you mean? As in?
Well, if I'm doing dynamic light scattering, for example, if I have TMV, then I have to go to Born and Huffman and pull out, you know, the appendix and get the, you know, the aspect ratios and stuff. But that's probably not a problem with what you do, right?
Yeah, exactly. So if you're talking kind of the static intensity profile, usually doesn't matter. It kind of falls out in the signal. So if you're talking core shell particles, and if you're talking kind of anisotropic non-spherical particles, usually because you're in DDM averaging over many, many particles, actually in the same way as DLS, you get in a kind of orientationally averaged setup.
So that sounds to me like the real advantage. I mean, the other stuff, you know, the size and stuff, that's all, we can easily deal with that. I mean, we can make small stuff or whatever, but that sounds like a really significant advantage for DDM.
Yeah. You've got some benefits. A lot of it falls out of the low Q, so the form factor just 1.
Yeah, and there's advantages for the dynamic light scattering, that if you go to high Q, then you can get rid of a lot of these problems, but you don't have to worry about the models if you go to low Q.
No, exactly. The model become quite nice. Another major advantage I didn't chat about much, though, especially when you compare it to DLS, is DDM handles turbidity so much better.
Well, we do that by going shallow penetration and backscatter, and then it deals with turbidity well too. But then you have to adjust the penetration. Yeah. Okay.
A lot of the times I'm talking a standard DLS setup. You might have a goniometer and be
Yeah,
able
no,
to do
this is a
multiple
special
angles.
version that we built. So-
Yeah.
Okay. Thank
So,
you.
Emma, you have a question?
Yeah, I have one question. Thank you. Lawrence, may I ask you a question about the resolution of the 3D holographic technique? Like, what's the distance that the technique can resolve? Like nanometers or tens of nanometers or hundreds of nanometers. For example, if I have a particle sitting just floating next to the substrate, I want to detect the distance of the particle to the substrate. So is that possible?
You are muted, Lawrence.
Thank you. Yeah, it's been a while since I've done that. So the limit really, I guess, is on how well you know where your substrate is sitting. So you can measure small motions in the particle position really quite accurately, and ultimately this comes down to the numerical aperture of your collection system in your optics. You can kind of dig into what that is. I mean, it depends a little bit on the type of lens you're using, immersion media, and all the rest of it. The minimum resolvable distance between two particles, certainly with a 500 nanometer diameter colloid, is something like 500 nanometers to a micron. So that's a kind of a rough level when you've got two particles close together. If you're talking about the absolute position in Z, we haven't hit the limit yet with the things that we're doing. I haven't pushed it that hard, but I suspect you could measure quite small changes as long as the particle properties are constant. So I would guess certainly hundreds of nanometers should be okay.
Hundreds of nanometers.
So Lawrence, if I may say, like generally your whatever streamline or movement of the motion is of the order of your particle size. So and your thing basically depends on the fringe size, like how much disturbance it is causing. So it should be of the order of your size of the particle or the things that you are resolving.
I think it should be smaller than that, because you can measure, if you're interested in measuring the centroid of something,
Mm-hmm.
if you're interested in measuring,
yeah. So Rayleigh criteria is lambda over 10, and Sparrow criteria lambda over 20. Yeah.
Yeah, for measuring the disk separation of 2 points,
Mm-hmm.
because you can do a little bit better if you measure it if you're fitting centroids. But it depends upon the problem, I guess.
Okay, absolutely.
So the objective. I'm very sorry to interrupt.
Yeah, go ahead. Please.
So may I ask, like, so we want to select objectives that have a very low depth of focus, and so that the resolution can be slightly improved, right?
Can I inject? I saw one of your objectives. You used an immersion objective. Can you talk about that versus an air objective? In one of your papers?
Yeah, sure. So we use an oil immersion objective in the early days when we were looking at, or if we're looking at small objects, because basically you get more fringes in your diffraction pattern, and that improves your axial resolution. So if you work with an immersion objective, that means you generally work, well, we generally work kind of close to the cover slip anyway, but it's to do with the number of diffraction fringes in your diffraction pattern, and the more of those you have, the better the axial resolution. So that was the reason why we went to high numerical aperture and immersion.
Diffraction patterns.
Was that a 63x oil? I saw it was a 60 something, but okay.
Yes, 60x oil.
I actually just realized there is a question. For example, we have particles moving in 3 dimensions, and is that possible to use the Jones technique to detect the diffusivity? Because I think Jones technique, DDM technique, is mostly like 2D technique from the presentation. So whether it's adaptable to 3D?
In short, yes. Your model becomes slightly different, but effectively, I'm pretty sure people have done DDM in 3D with some photo images. I believe Lawrence was saying, I'm not sure if you were using 3D DDM on your holography microscopies. I don't know at what point you were applying the algorithm.
Yeah, so the DDM processing, so I did a bit of DDM a few years ago. Actually, the averaging and the fitting, the modeling takes care of the 3-dimensional motions. There are some tricks you can play at high Q, because at the highest scattering vector, shortest length scale, your sensitive volume is actually very thin. So you can start to see particles disappearing in and out of the focal volume. But this is kind of, again, this is off into the weeds, so maybe we could talk offline.
But I meant your The example you showed where you combine DDM and DHM, were you using DDM on your 2D images or on your holographic stack?
Yeah, we just took the raw holographic images, fed them straight into the DDM. Yep.
Okay, I'm quite sure people have done 3D DDM. Because you can do 3D Fourier transforms, and it drops out nicely.
Thank you.
But Emma, if you are looking at like diffusion coefficient, like if you do it in 2D, I think that should be good enough to tell you what happens in the 3rd dimension, because these are small particles. So it does not, so they should be moving fairly the same way, right? If I'm not terribly mistaken. So that will be a good approximation until and unless you have like massive anisotropicity and things like those.
Yeah, we can like 2D model diffusivity. A 2D model it's like a 4D T equal to like a distance square. So 3D model is like 6D T because that like 6 directions equal to distance square. So there, in terms of modeling, there's some slight differences.
But I think his use is like the 6 that number, right? The Stokes-Einstein diffusive constant for DDM.
So effectively, yes, but the way you get the diffusion coefficient, so that's when I'm converting it. But you have to think carefully when you're doing the analysis. But yeah, they drops outright. You should get the same diffusion coefficient if you double it outright.
Anyway, so I think so, yeah. No, thank you. Our time is up. So thank you for thank you both Joe and Lawrence, both of you, and the listeners who listen to this. And again, so please keep on coming, inviting your friends to this, because this is, I think, a very informal way of learning about what others are doing. And as you can see, the conversation is more stimulating. You get to make more friends in the community in this way, I would say. So I will look forward to seeing all of you for the next talk, and you should have that calendar invite. We have a lot more interesting talks coming your way also. Okay.
Thanks a lot, Simon.
Thank you. Bye-bye.
Thank you.